I wanted to finish off sort of that thing and what's going on with the IgE, and it's really kind of an interesting process because here we can see you have an allergen that is present here. The antigen presenting cell is going to process it and present it to your type 2 helper T cell that's out there. Here we can see this type 2 helper T cell is releasing various interleukins to activate some B cells. These B cells right here are going to proliferate, and that's going to produce a whole bunch of plasma cells that are secreting that IgE. Unfortunately for people with allergies, the IgE loads up on mast cells in your skin right there, and this cell is sensitive now. And when this particular antigen comes along again, it doesn't have to get processed because you've already got the antibody ready to go, and its FAB is sticking off. So the allergen can actually bind this, and when it does bind, it's going to trigger that release of inflammatory molecules. Normally, this would work to destroy something like a parasitic worm. When the antibody bound to it, this cell could attack it, but in this case, you're sensitizing your cells in advance here. So your antibody-dependent cellular cytotoxicity is doing what it's supposed to. Unfortunately, it's doing it in the wrong situation here. And you're going to get this massive release of things like histamines and leukotrienes, and that's going to result in inflammation. So somebody who has allergies, even a really, really small percentage of antibody can be really, really powerful here. Just to show you sort of an example of one of these, I pulled this from chapter 19 where it shows you the skin prick test. And so they've taken various different cocktails of different allergens that are out there and effectively done a little skin prick on this patient right here. And yes, they do actually write in pen on the arm there. And what they're looking for is a raised area and then this surrounding redness. So a wheal and a flare, and that's going to tell you whether or not the patient is allergic to this particular allergen. You don't use a massive amount, and something like a bee sting could actually result in this person, if they are already hypersensitive, it could actually result in them going into anaphylactic shock and dying as a result of the skin prick test here. So you do have to be really careful and have your EpiPens ready to go. Sort of a summary, though, to put all this in perspective is to think about what's going on with the humoral immune response and your immune response in general. It's just really easy to show in this graph because we can monitor it really easily by looking at the antibody concentration in your serum. As this goes up, your B cells and plasma cells are coming into play. So we have our primary exposure right here, and you can see there's a lag built in. It takes about 10 days to get a good amount of antibody produced. And so if you have a really fast-moving pathogen, it could potentially kill you before your immune system can come into play. Remember, you've got your T cell independent activation of B cells. It's going to result in the secretion of IgM right there. And then you get isotype switching, and you switch over and make IgG. And think about it, IgM is going to agglutinate and neutralize, and your IgG is going to trigger all those different kinds of things. This is looking at serum, so you don't see an appreciable level of IgD or E or A. You would have to look in bodily secretions to see a good amount of A there. But you can see it takes a good 2 or 3 weeks before you start to have a significant amount of these antibodies coursing through your body to neutralize the stuff that's out there. If we switch and look at what happens after your second exposure right here, we can see it's a quicker exposure or a quicker response time here because you've already got your memory B cells in place. They're ready to go. They've gone through the clonal selection process, and you've got your memory cells hanging out with their B cell receptor already on the surface. That's going to trigger these guys to go through this process of becoming or differentiating into plasma cells, and they will start secreting IgM, but a massive amount of IgG, and you can see a much greater amount of that because remember, your IgG is the one that can do all these different things to boost your immune system responses there. Neutralize, but also trigger your white blood cells to start attacking and destroying these things here. And when you think about this, This is why we get booster shots over here. You get vaccinated, but maybe the memory B cells aren't going to be as much spread throughout your body. So you go in and you get a booster cell or a booster shot, and that's going to increase the amount of these memory cells. And that allows us to sort of segue into this last topic for chapter 18, where we look at acquired immunities. And if we look on the top, you can have natural immunities that are acquired that can be both passive and active mechanisms. If it's passive, you're looking at antibodies that are being passed through something like breast milk or through the placenta. Those antibodies will work in the fetus or the baby because they can trigger their innate immune defenses that are already in place, and the antibodies are going to actually facilitate that process, even though the antibodies came from mom. It's passive. You aren't actively mounting a response like you do in this one, where you actually go through the illness and you recover, and now you've got your active responses that are helping with that recovery, but you also have your memory cells that are there. We can do both of these processes through artificial means. And that's where we're looking at vaccinations down here. It's actively going to fight off the vaccine or the infection that is occurring there, and you're going to maintain memory of that later on. If we look at this top corner right here, we can have artificially acquired passive immunity because we can harvest antibodies from people or from animals and then separate those out and get an anti-serum. So it's literally the serum fraction of your blood. Clotting factors have been removed, cells have been removed, and you've got that haze colored sort of fluid that is extremely rich in antibodies that could be specific, especially if you're looking at a person who has already gone through the disease that is out there. So we'll take a look at this process and sort of look at the history of it. And it's kind of interesting. This shows you an example of what would happen if you got smallpox. This is opposed to syphilis, where you actually get great pox or pockets in your skin. These are small pockets, and they will appear on the palms of your hands and also the soles of your feet. So a little bit different from something like chicken pox that is out there. And the problem with this is if enough of these little pox sort of come in contact with each other, it actually causes your skin to sheet off. And if you lose that first layer of defense, pathogens can get into your body much easier, and you're actually going to die of dehydration. And remember, your gastrointestinal tract is really considered epidermal layer, and that can also peel off, and you will literally have a tube of intestines hanging out of your butt behind you there. And this practice known as variolation was actually discovered and started to be utilized in China, and you look at this, over 1,000 years ago. And what they found is that children who recovered from smallpox actually did not contract the disease a second time. And so they started taking some of this material from these scabs or pustules and grinding it up, and people would literally snort it. So imagine going to the doctor and getting vaccinated by snorting some scabs off of somebody who was sitting there in the doctor's office because they already had the disease there. This practice of variolation actually spread west through the Ottoman Empire, through Turkey, and eventually made its way to England and America in the 1700s. Eventually it was stopped because if you think about it, there's a lot of problem with cross-contamination. You have somebody who has smallpox, they also have syphilis or something else. And also if you got the wrong version of smallpox, there is two versions of the virus, major and minor strains. The major strain had 50% mortality. So there is a 50-50 chance you would die if you snorted that scab there. If we look in the 17 and 1800s, vaccinations started to come into play. And this is coming from an English doctor named Edward Jenner, who made the observation that milkmaids who developed cowpox infections did not actually get smallpox. And this is a cowpox lesion. Does not look much better, but it's less likely to kill you. And it turns out the cowpox virus is known as vaccinia, from the Latin for vacca, for cow. And this virus is actually very, very similar to our smallpox virus, known as variola virus. And the antigens on the surface of cowpox are similar enough to smallpox that your T cells would recognize cowpox, and now they were primed and ready to fight off smallpox. So you would get immunity, but you wouldn't get the nasty consequences of the smallpox disease. So really kind of a cool thing to use. And we do this because we want to develop what's known as herd immunity. And if you look at this process over here, you've got a few contagious people and a bunch of susceptible people, and that contagion spreads throughout this population because these people are not resistant. Well, what would happen if you had some way of developing that resistance in your population? And we can see that over here. This herd has immunity because these people have been vaccinated. And typically you want to be above 90% in order to cover a lot of the population so that you don't have an epidemic breaking out, because you've got fewer susceptible people in the population for that disease to spread effectively. And if we look at different kinds of data from this, you can see smallpox has been eradicated since they started vaccinating people worldwide. It's gone down, and the last known case of it was in 1977. The claim is that all smallpox has been eradicated, but we would have to trust the U.S. government and the Soviet Union to have gotten rid of it. And frankly, we can make smallpox in the lab because we already know the sequence. They're doing a similar thing now with the World Health Organization trying to eradicate polio because it is out there, and it's down to 3 countries so far. So we'll go through some of these differences between active immunities with vaccines and then passive immunity with immunotherapy. And if we look at the vaccines that are out there, an attenuated vaccine actually uses a live pathogen. So think virus or bacteria that has been weakened through some method. And the advantage to this is it's going to trigger your B and T cell response. But because it is live, it is difficult to store and transport. But the advantage to this is you get long-lasting immunity. The disadvantage, if you are immunocompromised, because it is a live strain, there is a chance that you can develop the disease, or there isn't a chance, there is a good chance that you will develop the disease. And also, it could revert, and this has actually been responsible for causing a poliovirus epidemic in the Dominican Republic there. Because it's live, some of these viruses can actually revert back to the pathogenic version. But the nice thing about live thing is we can give you a subclinical dose and you can infect the people around you because it is replicating, albeit weakly. If you look at a second type of vaccine that is out there, we can find inactivated or subunit vaccines. And these guys, you basically either break up the cells to give you your subunits, or you treat them with heat or chemicals. Think the aldehydes that cross-link things that are out there, or you could use radiation to kill these things off. And this is inactivating, or parts of the cells. And this is great because it's going to store easier. You also aren't going to get a severe active infection there. So you don't have to worry if you're immunocompromised or pregnant. But because it is destroyed a little bit, it's going to be weaker. It's typically going to trigger your B cells there, and we need to give higher doses and more frequent doses that are out there. The subunits, similar kinds of things. Multiple doses are required there. If we look at the last things, we can see toxoids, and those are basically bacterial toxins that have been inactivated. Again, heat or chemicals. It's going to trigger your B cells, and it does not prevent an infection because all you're doing is the ability to neutralize those toxins. But those are the ones that can kill you really easily. A conjugate vaccine is where you're taking polysaccharides and you glue them to a protein, and that's going to cause them to be endocytosed and processed so you can get your T cell response to this thing. And this is how they do a lot of vaccinations are conjugated, especially for things that you're going to use in kids to prevent things like meningitis out there. So if we look at this process here, again, think about this. Your vaccines are giving you your primary response to give you specificity, and your memory is around for when you're challenged with the actual pathogen that is out there. The last thing we can look at is artificial passive immunization. And here you're given an anti-serum that is going to neutralize and agglutinate things, and that'll decrease your disease symptoms quickly, but you don't retain memory of it. It's temporary but quick kinds of immunity out there. And if we look at all these different kinds of vaccines that are out there against all these different diseases, we are finding more and more of these cocktails of vaccines where they're putting multiple different types of antigens in these to give you different kinds of vaccinations. And it looks like DTaP and Tdap are doing the same thing, but it turns out one of these is actually going to be more effective at long-term immunity, and they're just now starting to look at this because they found kids who received the vaccinations with a newer version of it are less likely to have long-term immunity, and they're starting to see things like whooping cough from pertussis toxin. We're getting outbreaks of that. We might think we've got herd immunity, but it turns out the immune response works great with the vaccine, but it does not work great against the natural antigen that is out there. Then just sort of the last thing to summarize it, just to show you, this is our CDC recommended immunization schedule that's out there. And there are a lot of people out there that believe vaccines are resulting in increases in autism. That original paper, there's only one guy left who keeps saying it's true. Everybody else has said, I don't believe the data anymore. We've got multiple studies that were done in different countries, so you can't blame it on one government. Nobody has been able to show that the MMR, which uses attenuated viruses, and it does result in fevers because you do develop a little bit of a disease as a result of the attenuated viruses, that does not result in more autism cases when you compare kids who received MMR with kids who did not receive MMR. And in fact, a meta data study looking at all of these studies and putting everything together, the one thing they found is kids who are vaccinated are less likely to die of not only the infectious disease for the vaccine, but other infectious diseases too. So people that are out there that are saying vaccines can be harmful, yes, there is a chance that you could die from getting a vaccine, but you could also get hit walking through the parking lot by somebody who's on their cell phone. You could get bit by a shark and die if you're swimming out in the ocean, but people still do these things. Vaccinations are there to protect us. There is a chance that you might run into some problems. That is always a possibility, but if you don't get vaccinated, you are not studying for a test that might kill you. So hopefully you can appreciate all that's going on when we're looking at the host defenses that are out there, from your first line of defense with your physical barriers. And here you can see a pathogen entering through that first line of defense through the skin there. And the second line of defense with things like phagocytosis and inflammation, but also this third line of defense that is going to come into play to boost all of these non-specific defenses there. From your humoral immunity with your B cells and your plasma cells to your T cell response that is going to both destroy infected or cancerous cells, but also boost your immune system. And how we have this interplay between recognizing things inside the cells that are presented because you've got your antigen presenting cells to your B cells with the antibodies that are actually going to neutralize things and prevent them from spreading throughout the body. But remember, the overall thing that we're looking at with your immune system is this ability to both limit the spread but also destroy the infection so that you have specificity, and then you keep memory of that for later on so that you can start to fight it faster over here, but also start to influence the effectiveness of these non-specific defenses that are part of your immune system.